cancer patients can become weak and eventually die without obvious infection, metastasis, or organ failure, showing a symptom called placasia. Placasia, the major loss of fat tissue and skeletal muscle mass, is a significant factor in mortality rate in cancer patients. It's mainly associated with gastrointestinal and lung cancer, where the patients with these tumors experience the most weight loss with shorter survival time. While placasia may seem like the effects of food deprivation, nutritional supplements and drug-induced diet manipulation of diet does not reverse the effects. My goal is to study the pathway between cancer and placasia to better understand how placasia leads to cancer. To do this, I've overexpressed the oncogene in the intestinal stem cells to track the risk of cancer. An oncogene is basically a gene that has the potential to cause cancer, and the intestines are one of the fastest moving tissues. Its high cell turnover rate is a response to cell death and exfoliation due to the passage, digestion, and absorption of food. The intestinal stem cells also contain a gene called escargot that can be used as a marker to target gene expression, since this gene is only turned on during stem cells proliferation into daughter cells. So in the fruit fly, there's a system called the GAL4-UAS that can be used to direct gene expression. GAL4 is basically a transcription factor protein that can bind to the upstream activating sequence, UAS, that is similar to anti-cancer, which is used to target gene expression. So how this works is that if I have a fly with escargot GAL4 protein, and a fly with the UAS GFP DNA, GFP being the beta fluorescent protein that is, when expressed, fluoresces green under UV light, their progeny will have both the protein and the DNA, and GAL4 will bind to UAS, and GFP will be expressed in the intestines. This GAL4-UAS system can also be temporarily regulated, so the GAL80 protein can bind to the GAL4 protein, and under 30 degrees, this will inhibit GAL4 from binding to UAS, so GFP is not expressed. However, in higher temperatures, such as 30 degrees, the GAL80 structure changes so that it doesn't bind to GAL4 anymore, so GAL4 is now free to bind to UAS, and GFP is now expressed again. To find out which oncogene I wanted to use in the intestinal stem cells, I conducted a small experiment last semester where I crossed escargot GAL4, UAS GFP, tubular GAL80 temperature-sensitive flies with different UAS oncogene flies. This basically, the oncogene will be expressed in the intestinal stem cells in 30 degrees. It is known that fruit fly larvae and pupae have regenerative abilities. So in order to inhibit any developmental effects of on the oncogenic stress in intestinal stem cells, I shifted the progeny of these crosses to 30 degrees when they became adults, so the oncogene is only expressed when they were adults. And after I shifted them to 30 degrees, I started reporting their survival rate and found that RAS is most lethal in the intestinal stem cells of adult flies. A little bit background, RAS, many cancers arise from mutations in the RAS gene because it acts as a control that regulates cell growth and proliferation. When RAS is overexpressed, it's essentially always turned on, so cell growth and proliferation just keeps happening, which eventually can lead to cancer. My project is to overexpress RAS in the intestinal stem cells while inhibiting gene expression in the placasia-related tissue, such as fat bodies or muscle cells, using RNAi. RNAi is just a tool to inhibit gene expression. I will use flies with these two gene manipulation and conduct the same experiment I did earlier with the 30-degree adult flies to see if flies will be rescued or maybe effects of placasia will be lessened. If inhibiting a gene expression in the placasia-related tissue can rescue the fly, I would then study RAS in this gene to Gal4 UAS system alone does not allow for multiple gene manipulation. Any Gal4 driver can find to any UAS responder so that the gene expression happens everywhere. For example, if I had a fly with escargot Gal4, fat body Gal4, US RAS, and US RNAi, I would then have RAS expressed in the intestinal stem cells and fat body, while I have inhibit gene expression in the intestinal stem cells and the fat bodies too, which is not what I want. So to have dual expression, another system has to be used. The other system is called LEXA-LP. It works just like the Gal4 UAS system, except now LEXA is the transcription factor, the protein, and LEXA-LP is the enhancer, which looks like UAS. Gal4 UAS system has been around for a while compared to LEXA-LP. So it's possible to get the flies that work using the Gal4 UAS system just by ordering them. It's not the same for LEXA-LP flies. In order to get these flies, I have to clone the DNA I want, insert them into a plasmid, and send them out to a company to make the flies. Since I know for sure I want to overexpress RAS in the intestinal stem cells, I decided to do so using the LEXA-LP system. So to start on my project, I have to build the escargot LEXA line and the LEXA-LP RAS line first, which is what I did this summer with molecular cloning. So how that works is I cut out RAS gene from a plasmid that we had in the lab and the supposed area of the escargot enhancer. There's no definite way to tell where the enhancer for a gene is, but after looking at DNA databases, it seemed likely that it was upstream in front of the escargot gene. After trying to amplify different kilobases, different sizes of the DNA in front of the escargot gene, I was able to cut out a 2 kilobase area in front of it. So cut these genes out, and I inserted them into the respective plasmids. After checking to make sure that the fragments were inserted into the correct places, I sent these genes out to a company, it's called Best Gene, and they inserted the plasmids into the eggs of female flies. After many generations, they would send me back with a fly line that contains the escargot LEXA and another fly line that has LEXA-LP RAS. I recently received my LEXA-LP RAS fly back, and have not received my escargot LEXA fly back yet. But when I do, I have to check if the 2 kilobase area I did cut out does in fact contain an enhancer area by crossing those flies to LEXA-LP GFP. If their progeny does